Welcome back to the deep dive.
Our mission here is pretty straightforward. We take the sources you share, often dense scientific papers, and we really boil them down, get you the essential knowledge fast. And today we're tackling a big one, a really fundamental question in ecology and public health. What actually happens to the disease risk when we cut down forests? We're digging into a new paper. It's titled Using iDNA to Determine Impacts of Amazonian Deforestation on Leishmania Hosts, Vectors, and Their Interactions, led by Amy L. Massey and colleagues, just out in 2025. It really offers a new way to look at disease in these changing landscapes.
Yeah, it jumps right in, a long-standing debate, hasn't it? Whether
Yeah.
changing the land, like deforestation in the tropics, generally makes zoonotic disease risk go up, its amplification or down. The dilution effect is a huge question. Does more biodiversity dilute the risk because vectors bite lots of different, maybe less competent hosts? Or does disturbance wipe out diversity, leaving only the super spreader hosts and amplifying risk?
Right, and this isn't just academic. We're talking about places like the Amazon's arc of deforestation, huge forest loss, and critically, a heavy burden of diseases like leishmaniasis. This paper, Massey et al., tries to pin down the actual mechanisms. Their goal, essentially, was to use these really advanced molecular tools, this iDNA, invertebrate-derived DNA, to map out the whole network. The hosts, the vectors carrying the disease, the pathogen itself, see how it all shifts across the deforestation gradient. Okay, so let's unpack this study. Where did they even start?
Okay, location first. Really important. They worked in the southern Amazon near Sinop in Mato Grosso, Brazil. It's ground zero for the arc of deforestation. Rapid conversion of rainforest, mostly to agriculture. You end up with this complex mosaic landscape.
A mosaic. So,
Oh.
like patches of forest left within farmland.
Exactly. Patches of forest, big soybean fields, about 45% soy monoculture, and then pasture land too, maybe 11%. They specifically sampled across 39 different forest remnants.
And these remnants weren't all the same, right? They captured the whole range.
That's key. They picked sites representing a wide spectrum. Some were in areas still mostly forested, 100% forest cover within a couple of kilometers. Others were tiny fragments, down to only 13% forest cover in that buffer zone.
So they could really test how different levels of fragmentation impact things.
Precisely. But the really innovative part is how they sampled. This iDNA metabarcoding. It's like turning insects into little flying environmental samplers.
Okay, I love this concept. Mobile syringes, you called it earlier. Tell us more. What insects?
Right, two main types. First, and maybe most obviously, sandflies. They're the vectors for Leishmania. They trapped a huge number, over 56,000 sandflies. I mean, fieldwork alone.
Wow, 56,000.
Yeah, and they pooled them, groups of 50 flies per sample tip.
And from those pools, using the DNA, they could identify the sandfly species and what animals they'd been feeding on their blood meal.
Exactly, that's the power of iDNA. You get vector identity and host association from the same sample. But there's a catch. How do you know if changes in detected blood meals are because the hosts are changing in abundance or just because the sandflies changed their feeding preferences in different environments? Maybe they just started avoiding certain animals.
Right. The preference versus availability problem. So how did they control for that?
That's where the second insect group comes in. Carrion flies. They trap these separately. Carrion flies aren't biting hosts. They're attracted to scat and carcasses.
Okay.
So the DNA you get from them gives you an independent snapshot of the vertebrate community composition nearby. It's not biased by sandfly menus.
Brilliant. So if the carrion fly data shows the same pattern for host abundance as the sandfly blood meal data,
Then you can be much more confident that it reflects actual changes in host availability or local abundance in those fragmented spots. It's a really smart control.
it really is. Okay, so the target disease is leishmaniasis, caused by Leishmania parasites. What did the sandfly data itself tell them about the vectors present?
Well, they found 10 distinct sandfly taxa. 7 of those are already known vectors for zoonotic diseases. The most common confirmed vector genus was Leishmania. And another really abundant one, possibly a vector but not fully confirmed, was Setheromia uruguay.
And you mentioned RAI, Relative Abundance Index.
Yeah, just a simple metric, how often a particular species pops up in the samples compared to others. So Pei uruguay had an RAI of 0.87, really common. Leishmania, the main confirmed vector, was 0.79. So the important players were definitely there.
Okay, so the setup is clear. The methods are cutting edge. What did they actually find about deforestation's impact? Did sandfly numbers just plummet as trees disappeared? Surprisingly, no. The first finding was that the overall density of sandflies, all species lumped together, didn't really correlate significantly with the amount of forest cover.
Interesting. Not the simple story.
Not at all. But, and this is a big but, when they looked specifically at the medically important vectors, those Nisonia and P. argor, they found something else. Higher amounts of pasture cover strongly predicted fewer encounters with these key vectors.
Okay, wait. So not just general deforestation, but specifically conversion to pasture, areas with cattle seemed bad for the main vectors.
Exactly. It wasn't the soy fields, it was the grazing land.
Why? Any ideas?
Well, the paper speculates. Maybe the pasture environment is too dry, too open for the sandflies?
Yeah.
Desiccation.
Or maybe the cattle themselves act as a diversionary host. The flies bite cows instead of the usual smaller mammal hosts.
Could be. Or maybe it's insecticide use associated with cattle farming. That's a big unknown. But it flags pasture as somehow different and hostile to these specific vectors.
Fascinating. But you said the really big discovery wasn't about the flies themselves.
Right. The absolute core finding came from the blood meal data, what the flies were eating. They found that the probability of detecting a Leishmania host in a sandfly blood meal had this really strong nonlinear relationship with forest cover, specifically a quadratic relationship.
Okay, quadratic, so not a straight line. What does that look like?
Imagine a graph. Forest cover on the bottom axis, probability of detecting a host on the side. It's not that risk just goes up or down steadily. Instead, it forms an arc, like a hill.
Ah, so low risk in intact forests.
Low risk in heavily deforested areas.
But the highest probability of detecting these hosts was right there in the middle, at intermediate levels of forest fragmentation.
Precisely. That's the amplification zone, the messy middle, the fragmented edges. That's where the hosts seem to be most readily available to the sandflies.
Wow. And crucially, did the carrion fly data back this up? Was it really host availability?
It did. That independent survey from the carrion flies showed the exact same quadratic pattern for host detection.
Okay, that's convincing. So it wasn't just sandflies changing their diet. The hosts themselves, or at least evidence of them, were genuinely peaking in abundance or activity in those intermediately disturbed areas.
Exactly. It nails down that the mechanism is likely driven by host ecology in these fragmented landscapes.
So who were these hosts, the ones thriving in the middle ground?
The blood meal iDNA pointed strongly to two main players. Based on that relative abundance index, again, top hit, domestic dogs, Canis lupus familiaris, RAI of 0.22.
Domestic dogs. Okay. And the other?
Number 2, the 9-banded armadillo, Dasypus novum cinctus, RAI of 0.19, a well-known wild host.
And were these the same hosts showing up when Leishmania itself was present?
That's the clincher. They looked just at the sandfly tools that actually tested positive for Leishmania DNA, about 3.7% of them overall.
Okay.
And in those positive pools, guess which blood meals were most frequent?
Let me guess. Dogs and armadillos.
Bingo! Domestic dogs and 9-banded armadillos, it ties them directly to the pathogen in this specific landscape context.
So bringing this back to that big dilution versus amplification debate.
Yeah.
This study really doesn't support a simple universal dilution effect, does it? Where just having more species automatically lowers risk.
Not in this system, no. It seems much more about which species thrive when disturbance happens.
Right. These disturbance-tolerant species, the domestic dogs, obviously comfortable around humans, and resilient wild species like armadillos seem to do well, maybe even better, in these fragmented edge habitats. They concentrate there.
Creating that amplification zone.
Exactly. And it highlights that critical interface, doesn't it? Between domestic animals, like dogs living near people, and sylvatic or wild reservoirs, like the armadillos carrying Leishmania in the remaining forest patches.
That interface is where the spillover happens, where the disease cycle can jump between wildlife and the domestic sphere and potentially to humans.
Now, one important nuance about the pathogen itself, Leishmania. While the hosts peaked in the middle zone,
Yes.
the actual detection rate of Leishmania DNA, that 3.7% positive rate in the flies, didn't show a significant link to forest cover or pasture or anything they measured.
So the parasite itself can be transmitted across the whole gradient. Intact forest, fragmented, degraded.
It seems so. Transmission can happen anywhere in this system. But the probability of a vector biting a competent host, like a dog or armadillo, appears dramatically higher in those intermediate fragmented zones, because that's where the hosts are concentrated.
Okay, that clarifies it. It's about encounter rates. Looking back at the study overall, its big strength is clearly the scale and the insight from the iDNA, right? Linking vectors, hosts, landscape.
Absolutely. Doing this with traditional methods, trapping and identifying everything visually, would be monumental, maybe impossible at this scale. But there are limitations.
Like the pooling.
Exactly. Pooling those 50 sandflies per tube was necessary for cost and efficiency, but it means you lose fine-grained detail. You know, a fly in that pool bit a dog, but you can't build precise networks of which individual fly bit which individual host.
Right, you lose the one-to-one interaction data.
Yeah. And also, a general challenge with iDNA in places like the Amazon is the reference database. It's getting better constantly, but identifying every single bit of DNA relies on having a good, verified sequence library to match against, which is still incomplete for such hyper-diverse regions.
Makes sense. Okay, so let's distill this down. What's the big takeaway for, for you listening, for policymakers, for public health? Well, first off, thinking about disease risk in the Amazon, at least for Leishmania, forget simple linear ideas. It's not just less forest equals more risk.
Definitely no.
It's this complex quadratic thing, that intermediate level of fragmentation, the messy edge. That's the real hotspot. It's the sweet spot where key hosts, like domestic dogs and armadillos, seem to thrive, concentrating the potential for transmission.
And that really points towards needing targeted strategies, doesn't it? Focusing conservation and public health efforts, specifically on these edge environments, these transition zones, where domestic and wild systems overlap so intensely.
Yeah. And future research.
Oh, plenty to do. We need to understand why pasture is bad for those vectors. Is it the dryness? Is it insecticides? That needs pinning down.
And confirming the vector status of abundant species like Pauargai. Is it actually transmitting Leishmania effectively?
Critical question. If it is, given how common it is, that's a big deal.
So here's a final thought to leave you with. Given that domestic dogs emerged as such a key host, right there at the interface of forest and human settlement, what does that imply about the role of, say, veterinary public health? Things like managing dog populations, treating infected dogs, even just responsible pet ownership. How much could that influence human disease risks spilling out from these fragmented Amazonian edges? Something to think about. Thanks for joining us for this deep dive.